Hi everybody, today we're going to be talking about PCR and gel electrophoresis. But before we get started, I just want to do a quick recap over DNA replication.
The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand.
Recall there are some essential enzymes involved in DNA replication. Helicase is used to split and unwind our double-stranded DNA so that we can get a single-stranded DNA template. Primase is used to mark the starting point of replication through the placement of complementary primers. DNA polymerase is the enzyme that adds new complementary DNA nucleotides along the template strand of the DNA one at a time. Recall which nucleotides are complementary. Cytosine is going to be complementary to guanine, and thymine is complementary to adenine. Remember, DNA polymerase always builds in a 5' to 3' direction. That means that all nucleotides are going to be added to the 3' end. Polymerase chain reaction, or PCR, is a method used to make millions to billions of copies of a specific DNA sample. So this occurs through the heating and cooling of a few essential ingredients in a test tube. So PCR allows scientists to take a very small sample of DNA and amplify it to a large enough amount so that we can study it in better detail. So below we have a picture of 3 cycles of PCR. As you can see, after each cycle, the number of DNA fragments doubles. In just 30 cycles, we can produce 1 billion copies of the desired DNA fragment. There are a few required ingredients necessary for PCR to occur. So first, you're going to need the desired DNA fragment that you would like to replicate and put that in the test tube. Then you will need to add in DNA primers. So just like in normal DNA replication of an organism, complementary primers are needed to indicate the starting point for the production of our new DNA, or what we call DNA synthesis. In PCR, the experimenters are actually able to determine the region and the length of DNA that will be copied. They do this by choosing specific primers. The third ingredient that we will need is Taq polymerase. So Taq polymerase is just like any other DNA polymerases. So we said that DNA polymerase is used to synthesize new DNA by adding complementary nucleotides along the template strand, one at a time in the 5' to 3' direction. Taq polymerase does just that. The only difference is it is very heat stable. This makes it really important and ideal for PCR because we said that PCR requires heating and cooling, so we need something stable to withstand that. Finally, we will need to add in our DNA nucleotides to our test tube. So adenine, thymine, cytosine, and guanine, all of those are going to be added so that Taq polymerase can use them in order to make new DNA strands. There are 3 steps to PCR. At this point, we should have the desired DNA fragment, DNA primers, Taq polymerase, and nucleotides all inside of our test tube. The first step of PCR is known as denaturation. We've discussed denaturation before when we talked about enzymes and defined it as the alteration of a shape through some form of external stress. In this case, our external stress is going to come in the form of heat. When we heat the reaction, we denature the DNA, and our double-stranded DNA is going to become single-stranded. Rather than needing helicase to separate our strands like in normal DNA replication, in PCR we can just denature the DNA strands through the application of heat. The second step is called annealing. Here we are going to cool the reaction so that the complementary primers can bind to our now single-stranded template DNA. This marks the starting point for DNA synthesis. Notice that in PCR, no primase is needed. No enzyme is needed, just the primers. Finally, the third step is extension. Here, we raise the temperature of the reaction so that Taq polymerase, which functions very similarly to normal DNA polymerase, comes in and it can synthesize new DNA strands by adding complementary nucleotides one at a time to the 3' end. Let's say that you've just completed a PCR reaction. Now you have millions or billions copies of our target DNA. You want to check to see whether your PCR has worked. In order to do this, we can use a technique called gel electrophoresis in order to visualize and directly observe the fragments of DNA. Gel electrophoresis works by separating our DNA fragments based on their size and charge. In gel electrophoresis, DNA samples are loaded into wells at one end of the gel. Notice they are loaded at the negative end. An electric current is applied to pull the samples through the gel. Our DNA fragments that we just loaded into wells are negatively charged, so they're going to move towards the positive end. Our smaller fragments are going to move through the gel faster than the larger ones. Thus, DNA fragments are separated through the gel by size. The DNA fragments are stained, and the separated bands can be visualized. Here we can see DNA samples are loaded into the wells. One sample is placed in each well. When the power is turned on, an electric current is applied, and the DNA fragments migrate through the gel. The smaller fragments are able to move through the gel faster and further than the larger ones. Thus, smaller fragments will be located closer to the positive end. PCR and gel electrophoresis are used for many reasons, including paternity testing and forensics. Let's look at an example of PCR and gel electrophoresis at work. Samples of DNA were collected and amplified through the process of PCR. Then a gel electrophoresis was performed. Given the following gel electrophoresis results, whose DNA matches the DNA collected from the blood stain? If we compare the bands of DNA, we can see that the blood-stained DNA matches the DNA of John. I hope you found this video on PCR and gel electrophoresis helpful. Let me know if you have any questions and have a great day.